Oral examination from genetics. Number 1 question is, define genetics and its importance in fisheries. Genetics can help to improve fishery management. It is very important in fisheries in terms of breeding program. Efficient breeding program will be crucial to this development, not only to reach the production goal, but also to reduce production cost. Number 2 question. What is the chromosomal and chemical basis of heredity? Chromosomal is a structure found inside the nucleus of a cell. A chromosome is made up of proteins and DNA organized into genes. Number 3 questions. Explain the concept of population genetics. Population genetics is the branch of genetics that conducts the study of the genetic composition of populations. It combines genetics, evolution, natural selection, selective breeding, statistics, and mathematics. Number 4 questions. What's the importance of selection in fish stock management? The uncertainty in parameter estimates can be incorporated in stock assessment to produce a variable resource under a given management scheme. Number 5 questions. Differentiate hybrids from inbreeds and plan a good crossbreeding program. Hybridization is the process of crossing genetically different individuals to produce offspring, whereas inbreeding is the crossing of two closely related parents who share very similar alleles. Inbreeding is more likely to cause genetic abnormalities, unlike hybridizations. Number 6 question. Identify biotechnologies in fish genetics and determine how they could help increase productivity. Biotechnology allows scientists to identify and combine traits in fish and helps to increase productivity and improve quality. Number 7 questions. Differentiate mitosis from meiosis. Mitosis is a process of nuclear division in eukaryotic cells that occurs when a parent cell divides to produce two identical daughter cells. And meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. These cells are sex cells, sperm in males, eggs in females. During meiosis, one cell divides twice to form daughter cells. Number 8 questions. Differentiate DNA from RNA. DNA is a double-stranded molecule while RNA is a single-stranded molecule. Number 9 question. Discuss how and why DNA replicates. Replication occurs in three major steps: the opening of the double helix, separation of the DNA strands, the forming of template strand, and the assembly of the new DNA segment. Number 11 questions. Discuss how proteins amino acids are synthesized. During translation, ribosomal subunits assemble together like a sandwich on the strand of mRNA. They produce attract RNA molecules together to amino acids. Number 12 question. Explain the principles of YY male technology and the process on how to produce YY male produce by converting female into male population. Number 13 questions. Explain the different kinds of artificial selection methods. Mass selection, family selection, and multi-trait selection. Mass selection, this kind of artificial selection, which is the simplest form, is also known as individual selection. In mass selection, the best individuals are selected from a population for breeding and remaining individuals are discarded. The progeny of the selected individuals are usually better on average than the original population. Family selection is used when the heritability is small and or there are uncontrollable sources of environmental variances. Multiple trait selection. The best approach for selecting animals considering multiple traits is the use of an economic selection index. The overall multiple economically relevant traits by selecting animals using a selection index. Number 14 questions. Advantages of crossbreeding. Crossbreeding is used to produce superior fish for growth out, while selection is used to create superior brood fish. One of the most common breeding programs in aquaculture is the production of sex reverse brood stock to produce monosex population of for growth. Number 15 questions. Explain at least one topic in a fisheries biotechnology symposium and discuss the technology behind the topic.